staining technique. Staining is basically a process by which you are going to color the bacteria. As we discussed that bacteria are colorless organisms. Bacteria are colorless organism, and because it is colorless, and if we see on the microscope, we will not see the clearly because there is a background also. So it will basically it will going to वो बैकग्राउंड के साथ ही मिल जाएगा. तुम डिफरेंशिएट नहीं कर पाओगे कि व्हाट इज द बैक्टीरिया एंड व्हिच इज द बैकग्राउंड. इट विल बेसिकली mixed with the background. So in order to differentiate from the background, we are going to color the bacteria. So for coloring, we are using the staining technique. One technique we already discussed, that is called as the gram positive and gram negative, basically gram staining technique we already discussed. So if we're talking about the staining techniques, then it can be divided into the two groups. It can be divided into the two groups. The first one is called as the simple staining. Another one is called as the differential staining. For newcomers, these videos are on YouTube also. So if you want, you can take the reference. And after I will finish all the lectures for your exam preparations, you can pray for these lectures also. Okay. Fine. So first is the simple staining. Another one is the differential staining. The thing is, in the simple staining, we are using the one strain. Strain is the chemical. Strain is the chemical. Fine. So we are using the one strain. Okay. In the simple staining. In the differential staining, we are using the multiple strain. We are using the multiple strain. That is the first difference. Second thing is that in the simple staining, we are only able to see the size, okay, shape, all these things of a bacteria, and we can also differentiate. We can also differentiate between the gram positive, gram negative. Okay, that comes under the simple staining, and these are the strains. First one is called as the crystal violet. Second one is the sapphire. Is the counter strain. We discussed these both things in the gram staining technique. And another we have the methylene blue. It is used for the acid fast bacteria. It is used for the acid fast. What is the acid fast bacteria? It's the tuberculosis bacteria, TB bacteria. TB bacteria is the tuberculosis, you can say acid fast bacteria. Now coming to the differential staining, here as I said, we are using the different multiple strains. Okay, and what things we can see here, the first is the flagella, called as the flagellar staining. Okay, then bacteria, subbacteria have the capsule. So we are also seeing the capsule, that is called as the capsular staining. Capsular staining. Okay, and one thing is called as the negative staining. Negative staining. Now negative staining is we are using the different strategy. Here what we are doing, we are not coloring the bacteria. Instead of coloring the bacteria, we are coloring the background. We are coloring the background. तो अगर background को भी color कर दो, मान के चलो ये कौन सा color है, it's like a green in color, तो इस पे हम लोग yellow डालते हैं. ठीक, अगर येलो विजिबल नहीं है अच्छे से, तो इसको क्या करेंगे हम लोग, इसके लिए बैकग्राउंड हम इसको रेड कर देंगे, तो रेड पे येलो चमकेगा. आई मीन दिस लाइक विजिबल. फाइन. तो व्हाट वी आर डूइंग इन द negative staining is that we are coloring the background, not the organism. So this is all about your staining technique. Fine. And one thing more, you remember, we are also checking the endospores. Okay, we are also staining the endospore, that is called as the endospore staining. Okay, I'm not going in detail because this much is sufficient for your exam. Just remember gram staining. Why I teach you in detail? Because this is the frequently asked questions. So they may be asked again in examination, but you just keep the basic idea. If they will ask about the staining technique, then you start with that staining is a microbiological process by which we are coloring the bacteria in order to see on the microscope. Okay, that's how you define the staining technique. Then you classify this technique. Okay, and write down the description that in the simple staining, the one stain is used. In the differential staining, more than one stain is used. Simple staining for basically differentiating the size, shape. Okay, and whereas in the differential staining, we are checking the more deeper parts like the organelles of the cell. Okay, flagella, capsule. Okay, endospore. We discussed now what is the spore. It is basically a mode of sporulation is the mode of reproduction during the unfavorable situation. Bacteria produces the spore which is highly resistant, heat stable. Okay. So we are also coloring the endospore. Then what else? Yeah, one thing is called as the negative staining, where we are coloring the background, not the organism. Clear? So this constitutes this chapter. The chapter name is the general characteristics of microbes. It is a very, very important chapter. The only thing I can advise you to revise. Okay, if you revise, we will have the understanding. If you not revise, nothing will happen. And on the basis of this chapter only, you can deal all your chapters. Okay, this is the base. This is the base. Fine. You have any question, doubt? No? Anything you want to ask? Yes? You can ask. Yes? Transport media. So transport media, as I said you that we have the lots of basically the samples. Fine. So for preservation purpose, we are using the transport media. As the name suggests, no. In order to transport the sample to the you can say lab, it takes time. Fine. तो उसको हम लोग प्रिजर्व कर उसके अंदर का जो ऑर्गेनिज्म है उसको प्रिजर्व करने के लिए जो चीज़ हम लोग स्ट्रेटजी यूज करते हैं, जो मीडिया यूज करते हैं, that is called as the transport media. Example is the Struth media. Clear? Differential media to differentiate between the two organisms. Mixture of organism आते हैं हमारे पास, but we have to differentiate. In order to differentiate, अलग अलग चेक करना है, उसके लिए we are using the differential media. Example is your thioglycerate broth. Any other? No? So this chapter is finished. I am giving you the 2 days time. Read it thoroughly. We'll have the test. We'll have the test. The thing is, we want to grow the bacteria in a sterile condition. अगर हम लोग normal water लेते हैं, normal water may be containing the different you can say like the not good things, means maybe उसके अंदर में बैक्टीरिया हो सकता है, maybe उसके अंदर में कोई ऐसा केमिकल कंपाउंड होता है like the lead or anything, उसके अंदर में केमिकल कंपाउंड होता है जो इंटरफेयर करेगा बैक्टीरिया को ग्रो करने में. इसलिए डिस्टिल वाटर वी आर यूजिंग लाइक इट्स अ सैलाइन वाटर. डिस्टिल वाटर मीन्स सैलाइन वाटर. छोटा सा आता है, दवा सबके साथ भी आता है. दैट इज स्टेराइल वाटर एक्चुअली. ठीक है, उसके अंदर में कोई माइक्रोब्स नहीं होते हैं, तो हम लोग इंश्योर हो जाते हैं कि हाँ बैक्टीरिया के अंदर में अच्छे से ग्रो करेगा. फिर दूसरा अगर कोई होगा तो मे बी दे विल रिलीज सर्टेन केमिकल्स और वो जो हमारा स्टडी वाला बैक्टीरिया उसको मार ही दे. Okay, so we will not take the risks. We'll use the distal water. Any other questions? Yes. Sodium chloride क्यों डालते हैं? Sodium chloride basically for osmotic balance. Osmotic balance. NaCl used for the osmotic balance. Any other questions? Yes. No. Shall we stop the class? Yes. So thank you with having me. Bye. Have a nice day.